Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my session. So today we are going to talk about plant growth and development. It is lecture 3 of plant growth and development. Yes, thank you so much for joining my session, everyone. So Satyendra, hi, amazing world, hello, yes. So thank you for joining my session. So please do like, share and subscribe. Okay, so Satyendra has a doubt. So which says dominance is not an autonomous feature of a gene in case more than one phenotype influences by the same thing. Are you very sure about the last line? In case more than one phenotype is influenced by the same thing. Yes, so we will talk about what is codominance. So we will talk about what is pleiotropy. We will talk about what is polygenic. So you will understand this line. Okay, Satyendra? Yes? Fine. So I have not taught you what is polygenic genes, what is polygenic inheritance, what is pleiotropic gene. So once I teach, it will become easy to you. Okay? Yes? Alright, so today shall we go to today's lecture? So today we are going to talk about plant growth and development. So just give me a second. Yes, hi Dr. Merlin. Yes, a very happy good evening. Yes, Satyendra, a very happy good evening. So hi, Vishal is also here. A very happy good evening. Yes, Dharani, very happy good evening. I am good, thank you. How are you doing? Yes, thank you so much for joining my session today, guys. So please do like, share and subscribe. So do you have any doubt in the previous lectures of this chapter? Yes? Okay, so Dr. Merlin is Anthony. Okay, Anthony Abhi. Yes, hello Anthony Abhi. So thank you for joining my lecture. So please do like, share and subscribe. Who is this amazing world? I don't know. But Satyendra, guess the question that you asked me. Dominance is not an autonomous feature of a gene. So yes. So dominance is not an autonomous feature of a gene in case more than one phenotype is influenced by the same thing. So you have what is known as pleiotropic genes, where you have one gene affecting more than one phenotype. So their dominance is not autonomous. So autonomous is the one which is an independent body. So dominance is not an independent feature in case of more than one phenotype. Okay, so when a particular gene is controlling two different phenotypes, then the dominant is not an autonomous body or an independent body. So that is what it means. Okay, yes? Yes, so you have a doubt in relative growth. Okay, I will explain. So yes, Shimpi, hello, very happy good evening. So thank you, Shimpi, for joining my lecture. Yes, I'm good, thank you. So please do like, share and subscribe. Okay, so you asked me a question in Telegram before 3 days. Alright, so I'm sorry, by mistake I would have missed it. Okay, so I will check to it, Vishal. I'm really sorry. I will get back to you after class. Alright. Yes, so you have a doubt in relative growth rate. Yes, I will explain what is relative growth rate now. Yes, human skin color. You're absolutely right, Satyendra. Okay. So with this, today we move forward. So I will need today's lecture and one more lecture to finish this chapter. So probably on Saturday I will be done with this chapter. Okay. So today it's lecture 3 of plant growth and development. So before going into plant growth and development, I have an announcement. So you guys wanted a motivation session. So you said you are feeling very low. Ma'am, we do not have any confidence, any motivation. So yes, as promised, how to stay motivated. So please join me at 6 p.m. this evening to know about how to stay motivated and what is there. So what is the secret behind? So this is at 6 p.m. this evening. So that is after a break of 1 hour. So please join me for the same. It is at 6 p.m. How to stay motivated. Yes? Okay. So will you all join me? Yes? Okay, so this is my announcement that I wanted to give you. Okay, so moving forward, so lecture 3 of plant growth and development. So what is the thing that we were talking about? So we introduced, so I told you how plants grow in a sequential order. So how come flower does not come first, but it's your root and your stem that comes first, then you have your leaf, and then you have your flower. So how does the plant grow? Know the order. So then we defined what is growth. So it is an irreversible permanent increase in size of an organ or an individual. So that is known as growth. And plant growth is generally indeterminate because plants retain the ability to grow throughout their life. They do not stop at any point. So they have this ability to divide throughout their life. Okay. Then growth is measurable. So how do I measure the growth? So I am talking at the cellular level. So I can measure it in terms of protoplasm. But under the microscope, to count the cell itself is a tedious process. So how do I measure if the protoplast has grown or not? So I take some relative terms to understand, yes, there is growth that is happening, like the size, the cell size, the volume, the area, etc. So something like that, the cell number, the cell size, so all that are measurable features. So what are the phases of growth? So we have been talking about the 3 phases of growth. 3 phases of growth. So what are the 3 phases of growth? The first phase of growth is meristematic. So they are actively dividing cells, right? So they form your primary plant body. So meristematic cells, they have a distinct cytoplasm. They have a significant nucleus. So the cell wall is thin. So this is about your meristematic phase of growth. Okay. Yes, Antonia B, what is your doubt? Yes, you have a doubt? You want me to explain something once again? Okay, so this is the first phase. The next phase is elongation. So your cells elongate in length. So they increase in the width of the cell. So here you have increased vacuolation. Yes, so this is how in your elongation phase the cells are. Link of Telegram. So you didn't get the link of Telegram. So this link beneath my description box, so it is not working. Is that Telegram link not working, Antonia B? Okay, so I will ask them to repaste the Telegram link so that it is valid, so that you can still join the Telegram group. Okay, Antonia B? Yes. So did you solve the homework of yesterday? So yesterday I gave you homework in inheritance of one gene. So did you go back? Did you look at the questions? Was it easy? Were the questions tough? Yes, any feedback on that? So did you even open my PPT? So did you download it? No? Yes? Okay, so continuing. So this is the second phase. So the third phase is maturation. So this is when they lose the ability to divide and they take up a final function. So here you have plasmodesmatal, or your protoplast gets modified. So here you have abundant plasmodesmatal connections. So these are the phases of growth. Yes, no doubt. You solved only one question. Okay, so yes, so which means you have put in an effort to open the PPT, download the PPT, and you solve the question. That's okay, Shimpi. That's not bad. I really appreciate. So very good. So these are the 3 phases of growth. Then we were talking about growth rate. So what is growth rate? So the production or the increased growth per unit time. So whenever the term rate comes, so that is per unit time. Okay. Little thinking based questions. So which means even Anthony Abhi has tried. Okay. So how about the others? Okay, so now we are talking about growth rate. So rate is per unit time. So we spoke about 2 types of growth rate, right? So what are they? So EC genetic questions. Okay, so Satyendra found it very easy. Okay, fine, very good, not bad. Okay, so we spoke about two things. One is your arithmetic and the other one is geometric growth rate. So how about in arithmetic? So what is the example of arithmetic? Root elongation, right? So in arithmetic, so if a mother cell gives rise to two daughter cells, only one daughter cell retains the ability to divide, the other does not undergo division, right? So this is your arithmetic growth rate. So what is the formula? So LT is equal to L0 plus RT, right? So this is your arithmetic growth rate. I hope you remember. You did not solve, but you will do it today. All right, Vishal. Okay, so please do and get back to me so that you will have a proper understanding. Okay, anyways, in tomorrow's lecture, we'll be discussing the same. Okay, Vishal? Yes? Fine. Now, geometric growth rate. So, this is when a mother cell gives rise to two daughter cells and both the daughter cell continues to divide. So, this is your geometric growth rate. So, here the formula is W. You can say WT is equal to W naught E power RT. So, this is in terms of your geometric growth rate. Okay. So, how do I compare the growth of two living organisms? So, suppose I have two mango plants. So, I have mango plant A and mango plant B. Okay, so to look at, they are of the same height, say they are of this height, so they have leaves, etc. But per unit time, which is growing faster? So how do I compare the growth between these two mango plants? So how do I compare the growth between these two living systems? So I have two ways by which I can do. First thing I can do is absolute growth. The next one is relative growth. So Swami, you have a doubt in relative growth, right? So pay attention. So I will teach you with reference to my example. Suppose the first mark, so I am giving a test to all of you in plant growth and development, and the total marks of the questions, so the total marks of the exam is 100. Okay? Yes. So suppose Shimpi scores 87 marks out of 100. So how do I calculate her percentage? So how do I calculate her percentage? So it is 87 by 100 into 100. So this is with respect to the whole marks, the total marks I am doing. So this is absolute grading. So this is absolute comparison. Okay. The next one is relative. So though the total marks are 100, the first mark of Shimpi is 90, the second mark of Vishal is 85. So now, how do I calculate the percentage of Vishal? So I calculate the percentage of Vishal with respect to the first mark. So it will be 85 by 100 into 90. So with respect to the first mark I am doing, so it is relative. So this is known as relative grading. So similarly, if I want to compare the growth of two living systems, okay, so here when I say absolute, so I talk about the total growth of the plant. So this is total growth. Okay, the next one is relative growth. So this is with respect to the initial parameter. So this is with respect to the initial parameter. So Swami, did you get it? Did you understand what is relative growth and what is absolute growth? Yes. So still, do you have any doubts? So my voice is breaking. It's breaking in between. Okay. Now let me know if it breaks again. Yes, Shubham sir. Hello. Very happy evening. So thank you so much for joining our lecture. So please do like, share and subscribe. So is this clear? Absolute and relative growth rate? Yes? Okay, Swami, very good, done. So next thing that that we were talking about is the growth condition. So how should the plants grow? So what are the conditions needed for the plant to grow? Yes, of course, it needs oxygen, it needs water, and it also needs your nutrients. So water is the most limiting. So all the enzymes need water as a medium to perform its function. So this is water. Okay, so these were the conditions. So these are the points that we were talking in the previous lecture. So shall I go to today's concept? Yes, do you have any more doubts here? So should I explain anything once again? Yes, let me know. It's still breaking. Okay, so please give me a minute. Yes, please give me a minute. Okay, so they are checking. So let's. Okay, so Sikinder has a question. Consider a gene that has two. Okay, so this is the question that I asked in my homework questions yesterday. So can we discuss this tomorrow? Yes. So tomorrow in the lecture anyways, I'll be discussing all the 20 questions. So that time I will give you. Okay. What are the chances that they will once again? Yes. So I will solve exactly the same question tomorrow in class. So it was one of the homework questions, right? Yes. Okay. So I will solve it tomorrow. So we will finish it now. So it's good that you have tried. Okay, so moving forward to today's chapter. So today we are going to talk about a new topic, so that is differentiation. So what does differentiation mean? So when a meristematic tissue loses the ability to divide and takes up a permanent function, so that is known as differentiation. So it has differentiated to become something. So you have one mother cell. So when the mother cell continuously divides to give daughter cells, and one fine day the daughter cell says, I am going to become the hepatic cell, so I am going to take up the function of your liver. So that is known as differentiation. Okay, yes. So cells derived from root or shoot apical meristem and cambium differentiate and mature to perform special function. So that is known as differentiation. So cells which are derived from apical meristem, so which means after continuous division, so they become differentiated to take up a particular function. So this act of maturation is known as differentiation. So understand, you have a mother cell. So the mother cell continues to give rise to daughter cells. At one point this cell stops dividing and it undergoes maturation. So it takes up a mature function. So it is well developed, right? So it takes up a permanent function. So this act of maturation is known as differentiation, whereas this continues to further divide. So when it loses its ability to divide and takes up a final function or a permanent function, that act of maturation is known as differentiation. Okay? Yes? Okay, so they also differentiate, so it is unlike in maths, you don't have to write dx by dt, so don't worry Shimpi, okay? Yes sir, a very very happy good evening, so thank you so much for joining my lecture, so please do like, share and subscribe, alright? Yes, so if you have differentiation, so here they are continuously dividing, here they are taking up a function. So here the cellular changes happen. So you have major structural change in the cell wall and in the protoplasm, because when I said a meristematic tissue has a significant nucleus, has significant plasmodesmatal connections. So when it undergoes maturation, it has protoplasmic modifications. Alright. Yes, so it has structural changes in the cell wall and in the protoplasm, like relative growth rate. Okay, fine. So example is your tertiary elements. The cells would lose their protoplasm. So you remember your tracheids. Yes, so tracheids come in your xylem. So you have xylem vessels, tracheids, parenchyma and fiber. So out of this only parenchyma is living. Your tracheids lose their protoplasm and they become dead, right? But initially the tracheids are living. They contain your protoplast when they undergo meristematic division. But when they tend to take up a final function, they lose their protoplast. So that is why they become dead, because now they have taken up a final function. They are performing the function of transport of water, right? So this is known as differentiation, right? So they develop very strong, so they lose their protoplasm. Instead, they develop very strong elastic lignocellulosic secondary cell walls to carry water to long distances. So which means they are taking up a specialized function. Alright, yes. So this is differentiation. So initially what it is, so I have a cell, yes. So the cell undergoes division like this. Now it stops its ability to divide and it undergoes maturation. Okay, so it loses some protoplast, it becomes very thick, it contains lignocellulosic deposition. Okay, so which by result has become a permanent or a differentiated cell, right? So this process is known as differentiation, right? So when the cell which has lost its ability to divide again regains its ability to divide, again when it regains its ability to divide, so this is known as de-differentiation. You're understanding? So initially you have a cell, it is very meristematic, so it is undergoing division to form two daughter cells. So out of that, one daughter cell is saturated of division, so it undergoes some maturation and takes up a final function. Okay, yes. So in this function what happens, they have some protoplasmic modification. So example are your xylem tracheids. So this act is known as differentiation from the ability to lose the division capacity and take up a permanent function. So this is differentiation. But sometimes this differentiated cell can once again regain its ability to divide. That is known as de-differentiation. Okay. So now you understand what is differentiation and de-differentiation. Yes. Okay. Now this cell is once again capable of division. So once again it loses its ability to divide and it becomes a mature tissue or a permanent cell. So this process is known as re-differentiation. Done? Yes. So differentiation is the first time when it loses the ability to divide. Once again, if it regains the ability to divide, it is de-differentiation, and this, when it again loses the ability to divide, it is re-differentiation. So did you get the clear-cut distinction between these three terms? Yes. Is it clear? Yes. Is this clear to everyone? What is differentiation, de-differentiation and re-differentiation? So should I explain once again? Yes, let me know. Should I explain once again or is it clear to everyone? Yes, clear everyone? Alright. So living differentiated cells having lost the capacity to divide can again regain the capacity of division under certain condition. So that is known as de-differentiation. Alright. So formation of meristems, that is interfascicular cambium and core cambium from fully differentiated parenchyma cells. Okay. So understand. So you have your phloem. So you have your cambium, you have your xylem. So this is a vascular bundle, right? So this is your cambium layer. So this is your meristem. So this is your vascular cambium and this is a meristematic tissue. So this cambium is known as intrafascicular cambium. Right? So you remember this? Right? So intra is within. So within this vascular bundle you have a cambium. So this is intrafascicular cambium and this is the first time. Okay? So it is still meristematic. So this vascular cambium gives rise to secondary xylem and secondary phloem, right? Yes, so this is what gives rise to secondary xylem and secondary phloem. Now, when it is capable of secondary growth after giving rise to secondary phloem and secondary meristem, so it also forms a cambial ring, right? It also forms a cambial ring. So this is known as your interfascicular cambium. So initially in this place you only had parenchymatous cells, okay. So parenchymatous cells are permanent cells, they do not undergo any division. But this parenchymatous cells under certain condition, they have the ability to continue this ring, that is it forms your interfascicular cambium. So this is an example of dedifferentiation, okay. So this is an example of dedifferentiation. Alright, yes. So such meristems or tissues which are able to divide and produce cells once again and they once again lose their capacity to divide, that is known as redifferentiation. Now it is interfascicular cambium, so it is capable of division, but after some time this also loses the ability to divide, so it becomes redifferentiated, okay. Redifferentiation. So differentiation in plants happens in open cells or tissues arising out of the same meristem but produces different structures at maturity. Yes, so they arise from the same cambium cell but it produces different structures at maturity. Your vascular cambium, your cork cambium, but they are producing different structures at maturity, right? So thus in all plant, differentiation, dedifferentiation and re-differentiation is absolutely important, okay. Yes, is this clear? So final structure at maturity of the cells or tissue is determined by the location of the cells within. So where exactly the cells are present. So is this clear? What is dedifferentiation and re-differentiation, everyone? Yes, no doubt. So can I move forward? Yes, let me know. Okay. So example, cells positioned away from the root apical meristem differentiate into the root cap cells, right? So if you have the root cap like this, so this is the root apex. Here you have the meristematic region. So they all develop from the same meristematic region. So initially you have the meristematic region. The root cap is also made up of meristematic cell, but later it takes up a permanent function to protect the apex of the root. Alright? Yes? Fine. So moving further, so I hope I have explained the concept. Now talking about development. So long the name of the chapter is plant growth and development. So I was talking about growth, how to measure growth, so what are the phases of growth, etc. So one more thing that I forgot to say, in your geometry growth rate there are 4 phases or 3 phases of growth. So what are the 3 phases of growth in your geometry growth rate? Yes. So first is the lag phase, then it is log or exponential phase, and the last one is your stationary phase. So how is the graph? So initially it is a bit slow like this, it lags behind, and then it goes quickly and then it declines. So this is my lag phase, so this is my log phase, and this is my stationary phase. So the topic name being plant growth and development. So long I was talking about plant growth, the defining growth, the measures of growth. So in what terms it can be compared, what are the conditions for growth, etc. So now I am going to talk about development. So how does plant development take? Yes, growth happens. Yes, my primary plant growth happens with the help of primary meristem. So that is your meristem and your intercalary meristem. Yes, so then I have my secondary growth as well. So secondary growth happens through my lateral meristem. So this is especially my vascular till its old age. So that is known as development. Okay, so you have a meristematic cell. So it undergoes cell division. It can also undergo differentiation. Then it undergoes maturation. It becomes a mature cell and it dies. Right? So this can undergo redifferentiation, dedifferentiation, etc. Is there always dedifferentiation then redifferentiation? Yes. So when you have dedifferentiation, after some time it also loses the ability to divide. So it becomes redifferentiation. Okay, Shimpi? Yes? Fine. So moving further. So what is this plasticity? So plants follow different pathways in response to environment or phases of life. So that is form different kinds of structures. So for example, so when we were small, so what kind of cycle did you ride? So we all rode a tricycle, a cycle with 3 tires. So later when you grew up big, so you rode a normal cycle but with an extra stand to support you, right? And finally we all ride the proper cycle. So at each phase in life we grow up accordingly. So directly no child learns to ride the bigger cycle, right? So when you're 4 years old you don't learn to ride the bigger cycle. So in plants also each stage has different structure. Maybe the juvenile phase has a different leaf structure and the mature plant has a different leaf structure. So it depends upon the plant. Yes, Ayyappan Shaktivel, a very happy good evening. So thank you for joining my session. So please do like, share and subscribe. Okay, so this ability is called as plasticity. So in plants, in juvenile phase, in plants you have 3 phases, right? So what are the 3 phases? Germinating phase. Then you have the juvenile phase, or you can say the vegetative phase, when the vegetative parts of the plant are developing, that is the root, stem and the leaf, they are the vegetative parts. And finally they have the senescent phase. So this is the old age. So in germinating phase they have a different leaf, in juvenile phase they have a different structure, in a mature plant they have a different structure. Sorry, after juvenile you have reproductive also, so I forgot. So this is the mature plant. So here they might have a different leaf, here they might have a different leaf. So this ability of plants to produce different structures according to their phases of growth is known as plasticity. Okay? Yes, I am very good, Ayyappan Shaktivel. So thank you. How are you? Alright. So did you understand what is plasticity? Yes. So example, heterophyllal in cotton. So phyllal is leaf. So hetero is difference. In cotton, in coriander and in larkspur, the leaf structure differs from the juvenile phase and the mature phase of the plant. Okay, so in such plants, juvenile leaves of the plant are different in shape from those in the mature plants. So differences in the shape of leaf produced in water and in air. So this is one example. The next one is the difference in the shape. So this is the entire structure difference. So this is the difference in the shape of leaf produced in water and in air in case of your buttercup. So this also shows plasticity. Alright, yes. So I have a picture here. So can you see? So let me erase it. So can you see the structure? So in case of a juvenile plant, that is your vegetative phase, you can say the teen age of the plant. So this is the structure of the leaf. So this is the adult one. Okay. So this is your buttercup. So this is on land. Okay, but when it is initially growing in water, so this is the structure. So this ability of the plant to change its structure according to the phases of growth is known as plasticity. Yes, is this clear everyone? Yes, any more doubts? So can I move forward? Yes? Okay, so moving further. Yes, so development is growth plus differentiation gives development. So you know what is growth, you know now what is differentiation. That is, they are taking up a permanent function. So growth plus growth plus differentiation gives rise to development. Alright, so development is under intrinsic as well as extrinsic factor. So intrinsic is internal factor, extrinsic is external factors. Alright, so internal factors are within the plant, external factors are your environment. Okay, so intrinsic factors can be intracellular, so this is within the cell. So it can be the genetic problem of the plant, the genetic makeup of the plant, or intercellular. So this can be between the cells, between two cells. So it can be the chemicals that is produced by the plant. Example, your plant growth regulators. So they can also affect the development of the plant. Okay, so how about the extrinsic factor? So extrinsic factor is your light, temperature, water, oxygen, nutrition. Okay, so now we are going to talk about this intracellular factors, that is your plant growth regulators. Alright, so in plant growth regulators, what are the different categories we are going to talk? First, we are going to talk about characteristics. So how should a plant growth regulator be? Then who discovered it? So this discovery is pretty important because in statement based questions you have confusing statements. Alright, then physiological effects of plant growth regulators. So by now I guess you know what are the plant growth regulators. That is your auxin, your gibberellic acid, your abscisic acid, your cytokinin, then you have your ethylene, right? So these are your plant growth regulators. So what is the function of each? Ma'am, it is very confusing. Sometimes the answer is auxin, sometimes it is gibberellin, so don't worry. So first we will read about each of the particular plant growth regulator, and then I will tell you how to compare. For example, this enables the lateral growth. For example, this so let's see one thing. So this helps in the growth of the plant. Even your GA also helps in the growth of the plant. Ma'am, then if a question comes, how do I identify? So first I will teach you each of the properties separately, and whichever point is confusing, I have combined it as a tabular column so that you will be very clear under what condition it is auxin and under what condition it is gibberellic acid. Okay, so don't get confused. So these are your plant growth regulators. So these are the three subtopics we are going to talk under your plant growth regulators. Okay, so the first one is your PGR characteristic. So how should a plant growth regulator be? So number one, they are small simple molecules of diverse chemical composition. So all your plant growth regulators that I wrote in my previous slide, they are all not of the same type. They are all of different types. So they are small simple molecules of diverse chemical composition. So PGRs, they are also known as plant growth substances, plant hormones, or they are also known as phytohormones. So all of these names signifies your PGRs, that is plant growth regulators. So once again, plant growth regulators are also known as plant growth substances or plant hormones or your phytohormones. Okay, so the first of their type is indole containing compounds like your indole acetic acid. So this is your auxins. So auxins contain indole compounds in them. So this is the first type of your plant growth regulator. The next one is adenine derivatives. They contain N6 for furyl aminopurine. So this is your kinetin. So this is also known as cytokinin. Okay, yes, so where the one that contains indole compounds or auxins, the one that contains adenine residues or N6 for furyl aminopurine are your cytokinins. The third one is derivative of carotenoids. They are pigments, right? So that is your abscisic acid. So this is the third one. Then terpenes, so if the compounds or the plant growth regulators contains terpenes, just like you say it has an indole ring, it has a imidazole ring, it has an aromatic ring, so it has a terpene structure, so they are known as glycerolic acid. Okay, yes, and finally if your plant growth regulator is a gas, it is ethylene. Alright, yes, called as kinetin, which promotes plant growth. Okay, Vishal? Yes. What is indole compound? So I am not giving you the structure of indole, but let me show you. So yes, so this is your auxin structure, right? So this is my indole compound. So if I write a link like this, so this is my aromatic ring, right? So this is known as indole structure. So when they contain indole compounds, it is known as auxins. Okay, so when they contain a purine derivative, so purine we have read as your nitrogenous base. So when they contain a purine derivative, so it is cytokinin. So in glycerolins, it is your terpene derivative. So then you have your gas, that is your ethylene, right? So these are the different types of plant growth regulators. Okay, so please explain again on blackboard. Okay. So I am talking about plant growth regulators. So they are also known as plant growth substances, or they are also known as plant hormones, or phytohormones. Okay, Vishal? Yes? Fine. So now I am describing how should a plant growth regulator be. So this structures will not come for exam. So I showed you the structure so that you can understand. So structures will not come, Antonia B. Okay? Fine. So how should this plant growth regulators be? So they are small, simple molecules from different chemical background. So what are the different types of PGRs? The first one is indole containing compound. So indole ring. So it is a ring like your aromatic ring. So indole ring compound. So that is your auxin. So auxin is a type of plant growth regulator which consists of your indole ring. Okay. The next one is which contains adenine derivative. So where did we talk about your adenine? Yes. So your DNA is your genetic material. So this DNA is made up of nucleotides. Okay, so nucleotides are of two types. One is your nucleoside. So when a nucleoside binds to a phosphate group, it forms a nucleotide. So nucleosides include your sugar and your nitrogenous base. Okay, so this nitrogenous base is of two types. One is your purines, the other one is your pyrimidines. Okay, so purines are adenine and guanine, pyrimidines are cytosine and thymine. In case of DNA, in case of RNA, it is uracil. Okay. Ma'am, in Tamil Nadu within 1 month another method instead of NEET report will come. Okay, so we are waiting with fingers crossed. Let me know. Okay. So this is your nitrogenous base. So here they are containing adenine derivatives. Especially it is known as N6 for furyl amine derivatives. Okay, yes, and this is your kinetin or cytokinin. So this is the second type. First type, second type. The third type is your carotenoids. Okay. So your carotenoids are your pigments, they are your gibberellic acid. Then you have terpene containing, so that is your abscisic acid, and finally gases, that is your ethylene. Yes, is it done? Yes, Vishal, now you understood? Yes? Clear? Okay, fine. So carotenoid is okay, so I mixed up. So carotenoid is abscisic acid, so terpenes is gibberellic acid, I wrote the otherwise. So oxin is indole, kinetin is adenine derivative, your abscisic acid is your carotenoid, so remember Ca, okay, or then your terpenes is your gibberellic acid, so remember Tg, alright, yes. So these are the characters of your plant growth regulator. So I have tabulated here. So this plant growth regulators are of two categories. One is plant growth promoters, the other one is plant growth inhibitors. So your PGRs are broadly classified as plant growth promoters and plant growth inhibitors. Alright, yes. So promoters, they promote growth, right? So they are involved in growth promoting activity like cell division, then cell enlargement. So this is meristematic phase. So this is followed by your elongation phase. So it is easy to remember. So that is why I am writing. So then pattern formation. So it is differentiating here. So which type of cell it is going to become, xylem or phloem? So pattern differentiation, then tropic growth. So tropic growth is towards the growth of the sunlight. It becomes mature enough, then it undergoes the reproductive phase. So that is flowering. So plant growth regulators induce flowering. Then after flowering it is fruiting, then it is seed formation. So it is in order. So these are your plant growth promoters. They are involved in growth promoting activities like your cell division, cell elongation, pattern formation. You have your tropic growth, this is maturation, flowering, fruiting, seed formation. Okay, yes. Alright, Antonia B, that's okay. So who are your plant growth promoters among the 5 categories? Your auxin, gibberellin, cytokinins are your promoters. So totally 5 are there, right? So auxin, gibberellin, cytokines are your plant growth promoters. Abscisic acid and your ethylene are your plant growth inhibitors. Alright. So they play important role in response to wounds and stress of biotic and abiotic origin. So they are known as stress hormones, right? So Menki is on Saturday evening at 6:30 p.m. Okay, Ame? Yes? Okay, so they are involved in growth inhibiting activities. So this plant growth inhibitors have 2 functions. The first one is they play an important role in response to wounds and biotic and abiotic stresses. So number 2, they are involved in the growth inhibiting activities like dormancy. If they have an unfavorable condition, they go into a dormant period. Then abscission. So abscission is the falling of leaves. Then so what are the plant growth inhibitors? Abscisic acid and your ethylene gas. So gibberellins is denoted as Ga and abscisic acid is denoted as Aba. Right? So did you understand the difference between the 2? Yes? Yes, you're absolutely right, Shimpi. So is this clear? Yes, no doubts. So in the next class, we will talk about these 5 plant growth promoters as well as inhibitors. So with that, we will end the last lecture of this chapter. So yes, so discovery and the plant growth inhibitors and promoters we will start in the next class. So I will stop here. So please read your NCERT. So I am meeting you again this evening at 6 p.m. and we are talking about how to stay motivated. So please do join me and please do share the link with your friends. So until then, this is me signing off. So I am wishing you much and more. So stay safe and take care. So bye bye.